Hi. Hi, everyone. Thank you for being here. Thank you, Thomas, for the invitation. And thanks for the online audience as well to be there. Okay, so I will speak about the COVID-19 Host Genetic Initiative. And I will try to give you an overview of where we are, what we have learned in terms of host genetics of COVID-19. So how our genome impact the susceptibility and severity of the disease. And I will touch also upon maybe some progressing in long COVID as well. So the COVID-19 Host Genetic Initiative has started as a bottom-up collaborative effort back in March 2020. When I mean bottom-up, it means that really the community came together to try to work together to identify potential risk factor for COVID-19. It's been a fantastic experience and really showed the power of scientists coming together. We have not received any funding from granting agency or apply for any funding, just as an example of how you can be successful even without getting money from funding agency. The first idea of the initiative was to provide an environment where people could share results and find other people that could work together to answer research questions in this space. Later on in the development of this initiative, we started to be sort of a powerhouse for running a genome-wide association study and other type of association studies of COVID-19. So a resource that was really useful in the beginning of the pandemic, and I think myself I sort of underestimated, was the importance of tools to collect information from patients, but also informed consent and other material that could rapidly repurpose across different countries to rapidly start new studies. We have been working with countries with a traditionally underrepresented research. We've been working, for example, with Iran, with Syria, and with other places where some of these studies they would have otherwise take a lot of time. We had created a data dictionary with more than 200 variables. We've been working on communicate the research of other research outside English-speaking country, because science is very English-centered, but we wanted to reach a larger audience. So we had many translators translating the results of this initiative in different languages. Importantly, there was a catalog of studies. So we had more than 200 studies registered in our database, and you could go and contact a different study and start a research initiative. And we've seen some very high impact publication that actually came out by researchers that met through the platform and spare of some type of research related to host genetic of COVID-19. Most researchers are actually interested in this meta-analysis result, which means that we design study protocol that then are shared across the different studies, and then we take the result and put them together, for example, to identify new genes or variants that are associated with COVID-19. An important thing is that we make the result available before any publication or even before posting a result on bioRxiv. In fact, we have released in March, release 7, and we are now writing the preprint. This is not to say that you shouldn't go through a publication process, certainly very important, but in case of, if you want really result to be rapidly delivered in this context of emergency, probably the publication system is not the best system. We also had some silicon follow-up analysis, especially in the beginnings, where actually people will take the result, do analysis, and then put them directly online. Okay, so what we have learned over the past 2 years across on the host genetic of COVID-19, so why some people get infected, why some people get a severe COVID, what our genome tells us. And I'm going to do a relatively arbitrary distinction between rare variants and common variants, not because we expect the biology to be different between rare and common variants, but really because the technology is different. For rare variants, we use whole exome and whole genome sequencing. For common variants, we use GWAS array. So there is really just one robust genes that came out from rare variants association study was originally reported in 2020 using sort of a traditional Mendelian segregation analysis based on 4 individuals. It was a kind of very interesting clinical case. The gene is TLR7. And then since then, studies have started to increase in sample size and perform case control. More recently, we saw that in around 600 individuals and 1,000 individuals. And then as part of our, and this is mostly work done by others. And then as part of our initiative, we have conducted the largest exome-wide association study for rare variants in influenza COVID-19. This work was led by Guillaume. And you can see there is one gene that passed the sort of conservative exome-wide significant threshold, and this is TLR7, and clearly has a very large effect size, but these mutations are extremely rare in the population. This is an example on how some sort of candidate gene that were validated in a sort of more conservative setting when using strong statistical threshold, but there are many other genes that actually, unfortunately, has not been validated. So it's really important to think about. It's good to sort of put out hypotheses, but in the end, you will need to use this kind of technique to really being able to provide strong statistical confidence to your results. So from rare variants, the strongest result is TLR7. I will tell you a little bit later about the biology. What about common variants? So for common variants, we were much faster. We were faster because arrays are cheaper and because we are very accustomed in creating large consortia across many studies. There is a bunch of paper that you can read and about sort of what has been found for the human genetic of COVID-19. Interesting fact about the publication process, which might be relevant. We wanted to update our paper, but how do you do a paper that is constantly updated? It's not easy. That this sort of publication system is not ready for that. So we've been discussing with Nature. And so what we are doing is that we have our paper, this one here, and then on every new release of new result, we post another paper as a meta-arising, a sort of comment to the original paper. In this way, the reader goes to the original paper, and then he can see how the results have been evolving on increasing sample size. This is the first time actually the Nature used this approach, and I think it's an interesting way to think about sort of not putting just the result out there, but constantly keeping updating them. So in the last release, which is release 7, we have 82 studies from 35 countries. In the largest analysis, there are genetic information on more than 200,000 COVID-19 positive and around 3 million genotype individuals. Consider many studies, they span across multiple continents and multiple countries. You can see the cluster around Europe and US, but we actually had a good coverage of the Middle East as well. In Finland, we have been offering free genotype for our underrepresented countries, so we got a lot of sample from countries that otherwise couldn't afford genotyping. Nonetheless, we are still and remain very sort of skewed in the distribution of continental ancestry groups that are included in the analysis. You can see that the largest majority are Europeans. But as I mentioned, we have some representation from other ancestry groups from other countries, and I will show you why, at least in one example, why having other countries, other ancestries improve the power to discover new gene and new variants. Okay, so how do you do an analysis of COVID-19? What's the phenotype? It's clearly not easy because the pandemic has been changing, and one thing to disclose, and this is probably going to be our last release, is all these analyses were done with studies that mostly were collected before vaccination came into place. So we put aside a little bit the problem of vaccination, we still remain the problem on how you actually define critically ill, hospitalized, or sort of susceptibility for COVID-19. We found that the most powerful approach was actually compare these different groups with population controls. So in the first analysis, you have people that needed respiratory support, or they were in intensive care unit. And the second analysis, you have hospitalized COVID-19, and in the third analysis, you have everyone reporting any type of COVID infection. And as I mentioned, we use population control. Now, it's not a perfect design, right? Because someone in the population, now don't put yourself now everyone had COVID, so it's different. But back in the time, you could have, be a population control, meaning that you could become a severe case tomorrow if you would get infected. So there are clearly some potential misclassification. And so we did several sensitivity analyses using more refined definition of control, for example, antibody positives. And we found that the results replicate very well, but actually using these sort of rougher, more rough approach, it gives us more power. And that's why we use population control. So when we run this type of association analysis, we get this Manhattan plot. I don't know if you're familiar with them. Here, what you see here is millions of variants on the genomes with their chromosomal position. And here you have minus log 10 p-value. So higher peak means higher statistical significance. And then you have a line here that indicates that is the significance after multiple testing correction. I will speak mostly of two analyses, one called severity and the other susceptibility. And you can already appreciate, just by looking at that some peak change. For example, this one on chromosome 9 became much larger. When looking at susceptibility, this one on chromosome 3 became relatively smaller. There is another peak here popping up. So there are some differences. Now, we were interested from sort of more a methodological, but also biological perspective to separate genetic variants that were associated with susceptibility and those that were associated with severity. Now, this is an interesting problem because you cannot get a severe COVID if you don't get COVID in the first place. And so severity is always need to go through susceptibility. We use a Bayesian approach to sort of classify these variants in one of the two classes. And intuitively, a susceptibility SNPs or susceptibility variance is a variant where the effect on susceptibility is equal to the effect of severity, meaning that it doesn't influence any more the severity than it does on the susceptibility. So this can be an example of a susceptibility variance. A severity variance has still an effect on susceptibility, but has a much larger effect on severity. And so this is, for example, an example of severity variance. Now, if you plot sort of susceptibility effect size versus severity effect size, you see this sort of nice cluster. This was in release 7, in release 6. And then you can do some approach and give some posterior of these two to belong to one of the two clusters. If we do that, we have in total, this is the release 7, so the last one, we have 51 genome-wide significant loci. 9 of these loci are specific to susceptibility, or to be more correct, we can call it SARS-CoV-2 reported infection, and 35 have a significant effect on the severity analysis. For the other loci, we couldn't immediately cluster in one of the two groups. Okay, so this is sort of a little bit biological part of the talk, but I'm sort of, I'm gonna do a sort of an experiment. I'm gonna take a Manhattan plot here that you see here on the bottom, and these are all the peaks. Actually, you don't see my cursor there. I was moving my cursor all the time. So anyway, and I cannot move, so yeah, well, down there you can see the Manhattan plot. So you can see all these peaks. And then I took some biological pathways of COVID-19, from the viral entry to the recognition of the immune system and the immune response. These pathways were derived from orthogonal approaches, so not human genetic based. The genes that you see on these pathways are, for example, being candidated using molecular biology or immunology, or they are well-known pathways. Okay, so what I'm going to do is that I'm going to take the Manhattan plot and I'm going to see if I can map this peak on the Manhattan plot on these existing biological pathways. This will tell me that sort of by doing this sort of observational experiment or this genome-wide association study, I'm capturing known biology. So let's start with the virus entering in the upper respiratory tract. There it encountered the first airway defenses. One of the first things that the virus encounter when you enter in the lungs is mucus. And in particular, this MUC5B gene is a gel-forming protein that creates is involved in the creation of the mucus that protect the internal epithelium of the lung. Then the virus, if it passes this first the defense, progress, and it goes through the ciliated cell. The ciliated cell produce other sort of defense that are coded by these two other genes that are also mucins gene, MUC4 and MUC16, and they are involved in the clearing of the pathogen. The lung has an innate immune system, and it produces this surfactant protein D, which is produced by these FTPD genes. And this protein is involved in the recognition of the virus and in the regulation of the lung immune response. Now, the virus pass all these defenses, needs to attach to the cell. There are different receptors, the ACE2 receptor, but also the TMPRSS2 receptor, which is used to prime the S protein in the virus. And finally, the product of the LC6H20 genes, which is SIT1, interact with ACE2 to regulate the efficacy of the binding of the protein. So what is interesting is actually all these genes here, they tend to be candidated out of the, from this Manhattan plot, out of the susceptibility analysis. Now, the virus entering the cell, it needs to be recognized. There are several genes that work in the recognition. One is TLR7, which is the findings I mentioned before from the rare variants association. When the virus is recognized, interferons are produced, and interferons activate this STAT-JAK2 pathway, where there's many genes involved, the interferon gene and the JAK and TYK2 genes. The activation of this pathway recruit STAT1 and STAT2 from the cytoplasm, activated, send it back to the nucleus where bind to the genome and produce several genes, including OS1, which is a viral enzyme restrictor. And most of these genes, actually, they are coming out from the severity analysis. So something sort of beautiful is happening. We're taking a bunch of individuals, measuring their genome, taking a very rough phenotype. Have you been in the hospital for COVID or not? Unbiasedly comparing their genetic variants, and we see that we can sort of map these known biological pathways. For example, all these peaks that I mentioned map to what I shown you before. But what is probably more interesting is that if we get rid of this peak, there are many other peaks that reveal novel biology. And sort of this is the beauty of this hypothesis-free, unbiased screening. One of the strongest peaks is the one on chromosome 3, which is actually the largest signal for COVID-19, at least coming out from common variants. This came out just a few months after COVID-19. It is actually a quite interesting locus, because for much time we thought that there was only one signal, but then we figure out that there are actually two signals, one that is associated with severity and the other that is associated with susceptibility. And if you do some statistical conditional analysis, you actually see that the severity signal doesn't necessarily end up to the same gene that the susceptibility signal. So this one is in this gene called LZTFL1, and this is more here in this other gene. So this was missed by actually many studies, and was only an analysis done by Ancestry DNA that first found these two sort of independent signal. Now, what's happening in these logos? This is completely new biology. We didn't expect genes in this region to be necessarily implicated with COVID. So going back to that one, there's been several studies that have tried to figure out what's the underlying gene on this sort of region. There are multiple candidates. One is this ZTF1L1 that I mentioned. This group has done a lot in silico analysis to sort of support these genes. These regulate the epithelium mesenterium transition, or EMT, in the lung, which is done in response to inflammation and seems to have a role in viral control. Others as candidate is CXCR6, which is also somehow makes sense because it's cytokine related. A very interesting thing about this locus is that actually is or this haplotype actually underlying this locus, which is a very long haplotype, is its geographical distribution across the globe. Is almost unseen in Africa and is more common in Europe, but especially among Bangladeshi and Pakistani population. The reason why it's not seen in African population is because it lies on a segment of the genome that we inherited from introgression with Neanderthals. And so it's only common in population from the after the out of Africa bottleneck. Why is more common in Bangladeshi, Pakistani compared to European? We don't know. There's speculation that it may be a selective event in this specific population. But here's some data from Bangladeshi and Pakistani living in London. You can see that around 38% of Bangladeshi that are hospitalized carry these variants versus 20% that are antibody positive, but they're not hospitalized. Okay, so the biology is still a little bit unknown, but we can start to think about what are the sort of clinical impact of these genetic variants. Tomoko Nakanishi has led this work on where we have put together individual level data from around 12,000 COVID positive individuals with detailed clinical information. What she could find is that if you carry this haplotype, you have around 2.5 fold higher risk of being admitted to the intensive care unit. And this is around 1.6 times higher risk, even if you are hospitalized. So already even among hospitalized individuals, that increase the risk of even more severe COVID. We were curious to see if this sort of the comorbidity pattern we will observe in people having this haplotype and being hospitalized will tell us something about the biology. For example, there might be that these people will have more likely to also have hepatic injury or cardiovascular complication and so on. But actually, we didn't find that to be anything specific. It seems that people that carry this haplotype, they're just more severe in general, but it doesn't look to have a specific clinical trajectory. There is around 40% higher risk of dying if you carry this haplotype given you've been infected with COVID. And what is interesting is that there seems to be an age-specific effect. So what you see here is only individuals that have died or they have experienced severe respiratory failure due to COVID-19. And the two violin plot here, one are the carriers of the 3p21.31 locus or this haplotype. And here you have the one that are non-carrier. And you can clearly see that carriers tend to have a younger age. So they tend to have a severe COVID-19 or die for COVID-19 on a younger age than those that do not have it. The locus is also more predictive of COVID-19 at younger ages. So if you look at the bottom plot, you see all ages, and you see that in general, these risk factors, they are not, so what we are predicting here is the probability of developing a severe COVID-19, given you've been infected with COVID-19. And age and sex do a great job, already give you an AUC of 0.7, and other risk factors, they don't really add much on top of that AUC. When you restrict on individual younger than 60, you can see that you have a significant improvement by using this specific genetic information for this locus. So you go from 0.72 to 0.76. But what is important is that actually the effect is larger of other well-established risk factors, such as smoking, type 2 diabetes, and so on, and is similar to the effect of body mass index. This is not unexpected. We know that genetics has a larger effect in younger individuals simply because you have not been exposed to the environment long enough. And so you don't have many of these risk factors when you're young, and so they cannot predict. And the only thing you have left is your genome that doesn't go away. So that's the reason why it has a sort of a larger effect. We see these across many different diseases. Okay, so we got rid also of the peak on chromosome 3. There are 3 other peaks that I'm going to just describe because they carry some interesting biology. The first one is this variant on chromosome 19, DPP9. This variant is associated with interstitial lung disease, so increased risk of interstitial lung disease. I mentioned this example because we see a quite strong convergence between variants associated with COVID-19 and variants that have to do with pulmonary fibrosis or other disease like interstitial lung disease that are related to pulmonary fibrosis. So this indicates probably some shared biological effect. FOXP4 on chromosome 6 is quite interesting. This is one of the examples where having population or non-European ancestry population, it's valuable. The minor allele frequency is around 1% in European, but in East Asia is around 39%, and Hispanic Latino is 18%. And so we have enough statistical power to find these variants thanks to this other population. This variant is associated with lung cancer and interstitial lung disease again, and the gene is expressed in the proximal and distal airway epithelium. Similar example actually is ACE2, which clearly is a very strong biological candidate. We could find ACE2 only because the frequency of this variant that is just upstream of the promoter, it was higher in East Asians. So the last findings is ABO. The ABO, it's related to your blood type. And this one also been one of the first findings, both epidemiological and also from a genetic standpoint. It is associated with disease susceptibility, not with disease severity. This in the beginning was thought that was associated with severity, but is only associated with susceptibility. And in general, if you have a O blood group, you tend to be protected against the infection. Okay, so I'm spending the last few minutes speaking about long COVID. Okay, so COVID is probably, analysis on GWAS of COVID are probably over. It's impossible basically to study susceptibility. Everyone has had COVID. We don't have controls anymore. There might be some exceptional individuals that even being exposed, they never got infected. And the other is severity. We can still study severity probably by, looking at individual hospitalized, but for that we need very detailed vaccination information because clearly much of the cases that we see of severe COVID are unvaccinated individuals, and so that will confound the analysis. I would say that there are people still working on GWAS of COVID, but at least as an initiative, we're probably slowing down on that side. However, there are other interesting things we can do. One of these is long COVID. Now, long COVID, it's complicated. It's an heterogeneous condition, and the diagnostic criteria varies greatly across country, but also across physicians. For example, there are some physicians that do not believe simply in long COVID, and they don't diagnose it. So Vilma and Anna Ollila have started to put together different studies, many that have previously contributed to analysis of COVID-19 to study the genetic underlying long COVID. Now, genetics here can be valuable. I mean, I'm not going to say that genetic has revolutionized our understanding of COVID-19 because as I showed, there are some new biological findings, but basically we map to other pathways. And this is because we have so many years of experience in dealing with virus and in immunology. And so we could use many other orthogonal technique to understand the disease. But long COVID is actually one of these complex traits where we don't even know which tissue we should look for. And so genetics and this sort of unbiased screening might come to help when trying to understand the biology underlying this condition. In this analysis, we compare long COVID, and I will tell you later how we define that, with no long COVID. So we compare only among COVID-19 positive cases, but we also do a second analysis where we compare long COVID with population controls. Because these analyses use more recent studies, clearly we expect a much larger part of the population to be have been COVID positive. Long COVID is defined either via questionnaire or via diagnostic code. Most of the large epidemiological cohorts where genetic information are available, they have send out questionnaires, but clearly this is not scalable. We cannot, send out questionnaire to everyone. So this is only for specific research study, but this is still the best way to capture long COVID. For example, using symptoms that are not explained by alternate diagnosis or impact on everyday functioning for more than 3 months. The other way to get long COVID is through diagnostic code using the electronic health record, and this suits very well to big biobank study, which can link genetics to electronic health record. However, these codes has been released, I think, less than a year ago, and it's sort of underused. We have recently looked at the entire Finnish population, and I think it's been used only 4,000 times. And it's very, hospital and geography dependent, and so there's still some challenging on using these codes to diagnose of long COVID. So how the results look like? When we look at only COVID cases, we don't see anything. When we look at population control, we have a signal here. Actually, in FOXP4, if you remember, I mentioned it before. And this opened the question, are we gonna find out that the gene associated with long COVID are the same gene associated with COVID? Because we know that more severe COVID cases are more likely to develop long COVID, and so you might end up to just capture the same genes. What we found, however, is that actually, and you might get an intuition here, if you remember the plot for COVID, you have a huge peak here on chromosome 3, and here you don't see actually that peak. And when we compare the effect of gene associated with COVID severity with gene associated with the same effect for long COVID, we actually don't see a strong relationship, except for FOXP4. So this is very early data, and I don't want to make big conclusion out of that, but at least it seems that there are not necessarily a large share of effect between COVID severity and long COVID, and these specific signals seems to stand out. So in conclusion, our initiative, goal is to provide high confidence of association between genes and COVID-19, and that's what we try to do. We don't embellish too much our analysis. We do some sort of pipelining, and we try to send out robust results that can last the judgment of time and not turn out to be false positive later on. In the last analysis, we have around 50 loci. What surprised me when I start to work with this phenotype, I never work with any infectious diseases, so maybe to other people in the room, this might sound expected, but we find very different genetic effect on susceptibility and severity, so on these two aspects, and I would not have expected that. The result point to a clear biology, and it's kind of beautiful to see that you can take these statistical sort of results on a Manhattan plot and map them to biology so clearly. But you also reveal new unexpected biology, and this is really where the interest lies. This locus on chromosome 3 it's new. There's new biology to discover. As an effect of COVID severity in young individuals, is that comparable or greater to that of other established risk factor. Long COVID is a interesting and challenging phenotypes. We see one convergence signal with COVID severity, but not support widespread overlap. And I think the main message here is that working together as a community has really allowed for robust real-time replication on findings and exchange of results. There are many people involved. Actually, I think that we did the largest number of authors in any biomedicine paper. We had more than 2,000 authors. So you can go on this QR code and see everyone that has contributed. But really, the person, UI, is the person that has run most of the statistical analysis. So thank you very much.
Thanks a lot, Andrea. Great talk. And I invite questions. If you have questions in the room, please go up to the mic and test the mic, whether it's on.
Hello?
Yeah, very good. Go ahead.
Okay, so my question is that you discover about 50 loci is associated with COVID. Like, my question is that did you try to using those 50 discoveries to develop a risk score to see how can differentiate your case control? Did you try that?
Yes, there's actually a working group that is testing these. Yes, we have a polygeny score, and it does something, but nothing that is really clinically useful at this point.
Okay.
So it discriminates a little bit the severity. I mean, if you want an AUC, I think it is probably around below 0.6 or something like that.
Okay, so because I'm also working on COVID data, I got some data from UK Biobank, but we are looking at the gene-gene interactions. Basically, we are looking for pairwise associations. So I'm just wondering, do you think the 50 loci, because you just mentioned this project is kind of for your project you're thinking about it's finishing. Do you think it worth to take some, find some other method to looking for more variants, but not just the traditional GWAS study?
I think so. The only challenge is that, if you can use other methods on the same scale of data set as the one that we have used, there's definitely an advantage. Because the data access, unfortunately, is not so widespread, you will end up to, do new methods on smaller data set. And we know that sample size wins all the time. So a very simple additive model on a larger sample size is probably going to be more informative than every new model on a smaller sample size.
Oh, okay, then it comes to another question, how your data set is, because we try, I think it took us a while to get access to the UK Biobank. So I know you list there's maybe 200 studies from your website. So how easily for me, from outsiders, to get access to those genetic data?
Yeah, don't get me started. It's difficult. We are behind. We worked a lot with European individual level data for the chromosome 3. They're all deposited on EGA. The data access procedure is madness. You basically need to send an email to me, and then I give you emails of every PI, and then you have to send emails to every PI. I'm not going into details, but this call to me to sort of, we need a better data access sharing policies within Europe. I think US, it's a little bit better with DB gap. I know people don't like it, but yeah.
Please, could you continue after the session?
Sure,
Other people have questions too.
Sure, thank you.
How about you?
Yeah, thank you for the great talk. The severity of COVID also changes with the variant. So have you ever considered the sequence of the virus correlation with host genetics response?
Yeah, that's a very interesting question there. We should, unfortunately, there's no many data sets that have both information, the viral genome and the host genome. There's some smaller data set. People have looked into that. To my understanding, there's nothing so conclusive.
Okay. Have you ever sequenced the viral genome from the different patients with different severity?
Some studies have done that, but on smaller scale. I don't know of any clear signal of host viral interaction or something like that.
Okay, thank you.
But it's an interesting future.
I want to take
Yeah.
I want to take a question from the internet, from the virtual participation. And the question is, with these loci that you found now, do you feel like you have had a complete search, or do you think that there are other loci that you missed?
Yeah, we certainly miss loci. We know that if for every complex trait, if we increase the sample size, we're going to find more and more loci. So certainly there is a value in increasing the sample size.
Thank you. And there is a second question from the internet, and that is that there are ongoing studies on long COVID that may lead to differentiation, to defining subphenotypes of long COVID. So what is your take on that, and are you working on that?
Yeah, the long COVID working group is actually planning to do a symptom-specific analysis. Again, power and sample size here, it's an important consideration.
Thanks a lot. And there is one more question from the room, and that'll be the last one we take.
Thanks for the presentation. It was really exciting to watch. I remember the rare variant results you showed at the beginning were mainly identified in men. And this triggered the question, have you also done a sex stratified analysis? And did you find different results?
Yeah, so there are some paper on sex stratified effect. I don't know of any very robust sex-specific effect. The genetic correlation between males and females for most traits approximate 1. And I would not expect the COVID to be an exception about that. Where we see lower genetic correlation is things like testosterone, but also alcohol consumption and other things like that. But for sort of this viral infection, I would not expect much sex-specific effect, considering what we're seeing for other complex traits.
Yeah, I agree. Thank you.